session, Dr. Sean Toporek is going to be talking with us about cucurbit diseases, and he has a slide to kind of tell us a little bit about his background and story, so I'll let that cover our introduction.
Thank you. Well, thanks for coming to the plant pathology talk. I've got like half my trivia team from yesterday, so I appreciate you guys showing up and supporting me. It's really nice. So I am the new assistant professor of specialty crops pathology at South Dakota State University. So this is going to be a pathology-centric talk for you guys. I am from the Star, so I grew up between, Chicago and Milwaukee, which is why I sound like the way that I do. And then I went to Iowa State University for agronomy, got a degree in agronomy, not horticulture, and got introduced to plant pathology there. Kind of worked on my first pathosystem, which is Gauss's wilt of corn, just a bacterial streak. When I graduated, I went to Wisconsin, and I spent 2.5 years in a plant disease diagnostic clinic. So folks would send in their, suspected diseased plants, and we would diagnose them and try to give them some kind of recommendation. And then I also worked in a tissue culture lab certifying potato seed, cleaning up viruses as part of a larger seed certification program that Wisconsin has. So that was really cool. And then this looks a little weird, but I went down to Clemson then for graduate school, and this is where I got introduced to cucurbits. I spent 5 years working in cucurbit diseases, so I did a soil-borne pathogen of watermelons and a foliar disease of cantaloupe. And then when I graduated, moved to Colorado for a little bit, started working on perennial crops, worked on a perennial canker of peach for a bit. And then got this job. This job opened up right after, 11 months after I moved there, so I didn't really get to enjoy the state that much. But so now I am here. All right, so my new lab is going to be a specialty crop pathology lab. That means I'll work on the diseases of not only vegetables and your row crop vegetables, but also be working on tree fruit and fruit bushes as well. So there's going to be anything that is not essentially row crops I'm going to be doing a pathology program on. And so what I'm interested in doing is looking at pathogens, looking at their species, looking at their genetic diversity, their host range, their virulence, and then using that information to kind of prescribe management strategies that might be reflective of what I find. And then on the flip side, I'll look at our different management strategies that we use not to manage our diseases, looking at different cultivar choices, different host resistances we have, cultural practices, pesticide uses, and seeing how that influences the pathogen community and also our beneficial microbial community. So that's kind of my lab in a big nutshell. And then you may have seen that picture of what my lab actually looks like. It's just it looks like someone's renovating a kitchen. So I'm not really doing anything yet. I've only been here a month, so I'm just giving kind of a general talk here. All right, so in addition to that, I'm also the local foods education center director. So if you haven't heard of this center, it's like a 1.2 acre mock market garden that students run on campus. And so they plan, they implement, they harvest, and they donate all the produce that we collect from this farm we have on campus. And this is a good resource to do community outreach. I do some myself, but also train students how to kind of connect with the community, teach them about local food stuff. So we have 4-H kids coming through, FFA kids coming through, and we'll give them tours. In the future, kind of hope to make the local food center kind of a venue for local foods events. So we're going to have, kind of local food-centric movie nights. We might do a farmer's market, like a night market that kind of ties into other extension activities. And then I'd like to have some kind of grower talks of our own out there, which would be cool. Okay, and then I do not work for the diagnostic clinic, but I do want to plug their services because really trying to understand how to manage your pathogens is impossible if you don't know exactly what you're working with. It might seem like you have something, but you really have to do sometimes molecular diagnostics that has to be done in a lab. You need a microscope to really confirm what you're looking at. So yes, please do take a picture of this. They are happy to receive all your samples. They'll take photos as well, I'm sure. Connie Tandy's been there for quite some time, so she's very experienced. And a lot of times when you send something in, they will, they'll give you a printed report back, and it'll give you some recommendations on what you can do for that particular disease. So pretty much every state has a diagnostic clinic. I worked at the one in Wisconsin, so please utilize this resource. And so, having only been here a month and I've never seen a living plant in this state yet, I reached out to Connie and I said, well, what diseases are even in South Dakota? What causes disease on cucurbits? And so I had worked on them, so I felt most comfortable talking about cucurbits, but I got everything for the last 16 years from her, and I've kind of categorized the 14 different diseases that were reported into these different categories. So there's fungal pathogens, there's bacterial pathogens, and there's viral pathogens. So I kind of grouped them, the fungal pathogens that spread through the air, the fungal pathogens that live in our soil, the bacterial pathogens that are seed-borne, bacterial pathogens that are vectored by insects, and then viral pathogens which are also vectored by insects. And this, to me, is a good way to kind of give you an overhead explanation of kind of how a plant pathologist thinks about managing disease, because really you can sort of group these together and think about them the same. So if you guys have not gotten tired of seeing this in everyone's slides yet, this is the plant disease triangle. And so it's just kind of a plant pathology 101 concept. You need your susceptible host. You need your virulent pathogen, so not just a pathogen, but one that's able to cause disease. And you need a conducive environment, which in plant pathology, 99% of the time just means some water and humidity. But I just wanted to emphasize that disease is an exception, not a rule. There is a lot of pathogen inoculum floating through the air all the time, but it's only in these kind of Goldilocks situations of plant stress, proper environment of your pathogen being there that you actually do get disease. So it doesn't always happen, despite all the opportunities it can happen. And so this is kind of a pared down what's called a disease cycle. So every pathogen you ever see in your field is going to have its own disease cycle. And it's important to understand this, at least from my point of view, because this is how you decide what kind of management strategies are going to work. And let's just start up here. Do I have a laser? No, that turned it off. Oh no. Okay. Is this a laser? That's the laser. Okay. So let's start at inoculation. Let's just say this is some spores floating around in the air. It's going to come into contact with your plant. It's going to infect it. You're going to get disease. And then it's going to reproduce. And so depending on your pathogen, it's either going to enter what's called a primary disease cycle or a secondary disease cycle. So this primary disease cycle is this outer one, and this inside one's a secondary disease cycle. So for some pathogens, they cause the disease, the plant dies, and then that's it. That's the end of the season. They go into what's called overwintering, and they'll start making long-term survival structures. They'll, burrow into the ground, something like that, and then in the spring, they'll germinate and start being able to infect your plants again. Other pathogens, before they overwinter, actually undergo multiple generations and produce more of themselves, and that's going to actually help spread them throughout your field. So knowing whether or not your pathogen has a primary only or a secondary disease cycle is going to be really important when you think about management. And so as plant pathologists, of course, we want to prevent inoculation, and that's going to stop pathogen reproduction. So let's look at fungal aerial diseases. So what I found from Connie's reports was that we had alternaria leaf blight, Cercospora leaf spot, powdery mildew, which I'm sure you've all seen, gummy stem blight, and cucurbit downy mildew. Those were the 5 reported over these years. And so here's what they look like. Alternaria leaf blight is kind of a light brown spotting, and it kind of looks like someone's sort of tracing inside your leaves smaller versions of itself. So it's kind of like if you draw a circle and you draw another circle inside of it, another circle inside of it. That's very diagnostic for alternaria leaf blight. Cercospora leaf spot just has these kind of tan little spots, but what's really diagnostic are these yellow halos that will form around those spots, right around here. Cucurbit downy mildew is almost kind of not worth showing because depending on what cucurbit it infects, the symptoms are going to look entirely different. So this is on a cucumber, which is very angular and blocky, but this disease does not look like that on a squash. So that one's tricky. Gummy stem blight are these dark lesions. Oftentimes it can be like V-shaped and often in the lobes of leaves like that, and they're always going to be accompanied by some stem blight, some gummy stem blight. And this is because of the gummosis that's going to come out of the stems. So I almost want to de-emphasize these two. So these are the ones that I studied. Well, I studied this one in South Carolina, and then my lab studied this one. And these are typically more southern pathogens. So I'm going to ignore them because I don't expect that you'll see them much. I was actually very surprised that they were even reported in this state. That was completely new news to me. It was just kind of in these internal documents that were in the diagnostic clinic. But of course, you're going to see cucurbit powdery mildew forever and for the rest of your life, especially because South Dakota is dry. And cucurbit powdery mildew, although it can do better in humid environments, is one of the few fungal pathogens that does well in a dry climate. And so we want to manage our foliar diseases because, you lose that photosynthetic area when your leaf starts to become necrotic. When, you get these leaf spots, you're losing photosynthetic area, and that's just going to reduce your yield because there's less, starches and sugars going into your fruit. But you're also losing leaf coverage, and that's going to result in sun scalding. And so, whenever I went through my research plots, I would kind of move leaves over just so I didn't get any of this sun scalding happening. So here is a disease cycle, and I understand that these are beets and not cucurbits, but I went to the internet and I downloaded these photos. I didn't have time to draw disease cycles for all the things I want to show you. Don't worry about any of the fungal names. That's just a distraction from the main point. But I want to start and go through this disease cycle with you guys. So here are your spores flying through the air. They come into contact with your healthy plant. They infect, they cause disease, and then this has a secondary disease cycle. So on your diseased plants, more spores are going to be produced. And those are going to blow to another healthy plant. Then more spores are going to be produced. And this is going to happen over and over and over again until the winter stops this secondary disease cycle from happening. And then it's going to start overwintering. And a lot of times it can overwinter in infested crop residue. It could survive in seeds. And then these two things are just examples of what are called overwintering structures. So fungi will produce these, and they can survive really wet climates, really dry climates. They're very physically tough. You can't destroy them. And these can last in your soil for years and years. And so let's start thinking about how we want to manage based on this disease cycle. So this is what's in our soil. So I know tillage is a rough topic in South Dakota just because you guys have like sustained 60-mile-an-hour winds at all time. And so you want to keep that planted, but crop debris is filled with all of these reproductive structures, right? It's this organism living in your crop debris. So if you can till that crop debris into your soil, you're putting it in contact with the other beneficial microbes in your soil, and that will outcompete them, and that will actually kill the pathogen that's on that debris. But if you leave it on the soil surface, it's not being competed against at all. So another option could be just, make sure you remove everything, but even like the tiniest little plant pieces are going to have these reproductive structures. Of course, use disease-free seed. A lot of the seed you buy should be disease-free or get your money back. Okay, so now let's say this inoculum is floating in the air, and before it hits your plant, you want to consider protectant fungicides. So protective fungicides are applied so that when the inoculum lands on your leaf, it's not able to cause that infection. It's killed before that infection process can happen. Host resistance is amazing if you can get it for your particular disease. That would always be my recommendation, is find the host resistance for the disease that you're having in your field. And crop rotation, which I will have a slide on after this, may or may not work. So rotating away from, rotating your crop away to something that your pathogen cannot infect biologically. So let's say that the inoculum is now on your leaves. What can you do? So systemic fungicides. So you can spray your fungicides and try to kill this inoculum before it actually establishes that infection. And then one thing I always like to recommend is that I understand, people like to intensively plant. If you're working on like an acre or two, you want to get as much in your field as possible. But what you're doing is increasing the humidity in your plots. And fungi love humid environments. So if you increase your plant spacing and maybe orient it with the predominant winds, you're going to be able to naturally dry off your plants after rains, and that's going to reduce the amount of successful infections that can occur from your pathogen inoculum. And kind of in the same reason, so if you have this diseased field now, try to stay away from overhead watering as much as you can. Always try to water the roots, not the leaves, because any rain splash on your infected plants is just going to splash it around. It's going to splash around these spores, and it's just going to spread to your other plants.
Now, if you did till any infested crop residue or got a little bit tilled in,
Yeah.
how many years would it be in the soil till it'd be safe to plant there again?
That depends on what disease you're talking about, and it's hard to know. I mean, it's like someone's got to go in there, they have to look and see, you know, how much, like they can look at these and see how many of those are in there. They can estimate like the amount of inoculum in the soil, and then they can actually do what's called it's called colony forming unit testing, where you actually look at your soil, you plate it out, and you see how many colonies grow out of that soil, and that gives you kind of an understanding of how, you know, alive your pathogen inoculum is. So it's really hard to kind of estimate that. But so it varies by pathogen, and it's just kind of really has someone put the work in to actually know that number is usually the issue. So here's the caveat. So you do want to do crop rotation for fungal pathogens that blow in the wind, right? But for instance, the pathogen I worked with, Cucurbit downy mildew, the spores of that, the pathogen causes Cucurbit downy mildew, but the spores of that disease start in Florida, and they will travel thousands of miles to a field up in Ohio, because these spores are what's called melanized. So they're actually pigmented, and they can survive, you know, exposure to UV radiation. So you rotating or you burying the inoculum for some of these fungal pathogens that can spread like this may not be effective, because as long as the wind's blowing and as long as people in Florida are still planting cucurbits, you're not going to be able to avoid, you know, this disease in particular. But now let's talk about soil-borne fungi, because they behave completely differently. And so Pythium root rot and stem rot, Fusarium root rot and crown rot, Rhizoctonia root rot, if you showed me some roots from these you know, infected roots caused by these diseases, I probably would not be able to differentiate them. I think Fusarium root and crown rot, you can see kind of like more red, reddish discoloration in your roots. Pythium, I think, infects kind of the fibrous roots a little bit more. But to the untrained eye, I would always send your roots into a diagnostic clinic to figure out what is actually causing the disease, because they will try to actually extract the pathogen from your infected roots, and they can actually tell you. Fusarium crown rot, though, this is kind of an exception. A lot of times you will not see this extensive rotting from Rhizoctonia and Pythium as you will with Fusarium. So this is one that you could be confident is Fusarium root rot if you do see that. So in addition to kind of like a mature plant root rot, you'll get damping off. So these pathogens will be in like your potting mix. And so when you go to propagate your plants, you know, you'll get various ages of plants coming up here and just dying off right away, you know, kind of getting cut off right at the soil line. And then the seeds themselves can rot even before they emerge. And that's because you're, you know, unintentionally planting your seeds into infested soil. In addition, the same pathogens that cause everything I just showed you also rot your fruit. So these pathogens are living in your soil. So in addition to being able to rot your roots, if you have fruit laying on the bare soil and they're, you know, kind of soggy or they're wounded, those fungi can actually come out of the soil and start colonizing your fruit like this. So I saw a lot of that in South Carolina. I don't know what I'll see here in South Dakota, but these are all possibilities. So I purposely wanted to leave this very complicated life cycle in here. One, just so you can appreciate how complicated fungi can be. There's a lot of steps along the way that happen in their disease cycles, but also because this is for Pythium root rot, the one that I worked on my master's, and there's kind of an extra little bonus I want to show you in this one. But this will be kind of emblematic of how soil-borne pathogens work kind of in general. So I'll start again up here at infection. So this is just saying I have some inoculum in the soil from my fungi. Put my plant is next to that inoculum, so now my seed's rotting, or I'm getting damping off in a seedling, or my mature plant roots are starting to rot, however this disease manifests. So this is just showing hyphae kind of growing in roots here, and then this is just showing like the lower stem of a plant and the roots, how it's all kind of colonized by these organisms. And then at the end of the day, you have a dead plant. And so this only has a primary disease cycle. So there's no secondary infections that happen from these soil-borne pathogens. All this is showing right here is just the crazy biology of the organism. And so you have this dead plant, and the organism's gonna be like, oh, okay, the season's over. I need to make some kind of overwintering structure. I need to survive until next year. So in the case of Pythium, it makes two different kinds. I won't even mention their names or anything like that. But these are the things that you need to worry about. This is what's in your potting mix. This is what's sitting in your field year after year, is these survival structures that allow this pathogen to hold over. So this is really where crop rotation works. So moving your plants out of that infested soil into a neighboring plot down the road, wherever you can go, is going to be one of your more effective methods of control. You can use systemic fungicides, but I don't think those really work that well in the field. Based off my experience, you can apply some of these things in the drip with your transplants, but really what you want to do is really use these fungicides for soil-borne pathogens kind of at the transplant stage. So if you're transplanting, you want to drench your trays with these fungicides. So that's going to be the most effective use of your fungicide money. And then there's other ways you can kind of try to remediate your soil fumigation, anaerobic soil disinfestation, soil solarization. You're going to have varying levels of efficacy on that. But those are all in an attempt to reduce the amount of these things that are living in your soil that can just stay there unless you manage them. So that's true for Rhizoctonia and Fusarium, and this is kind of where the story would end. And so when you plant your plants, these survival structures would kind of germinate in the presence of your plant roots or your seeds, because they release these exudates. Yeah.
So when you're crop rotation and you're moving new plants, they have to be of a different family?
They should be, yes, that's a good rule of thumb. I don't know if that always matters, depending on what it's. I know I've kind of lumped a lot of pathogens into one, but yeah, that's just a good rule to follow if you're not sure. Kind of switch up the crop families. I don't think it would matter for Pythium, for example, but it would matter for Fusarium. Yes, for sure. Sorry to kind of have the wishy-washy answer, but I've got but Pythium now, and you'll see this a lot in kind of flooded soils or poorly drained soils, is they actually have this part of their life cycle that is aquatic. So from these survival structures, they differentiate into these kind of swimming zoospores, is what they're called, and they have flagella on them. And so when you have poorly drained soil, these water films are created, within your soil, and those root exudates coming from your roots and your seeds will travel through those water films, and these guys can sense them, and they swim through those water films and find your roots and find your seeds. So it's just like this kind of added evolutionary step that this organism has over some of the other soil-borne pathogens. So drainage, improving soil drainage is always key to managing soil-borne pathogens. Okay, now
Well, if those are broadleaf diseases and you rotate the grass, that would be a really good rotation.
yes, that would be a good rotation. I don't want to say that 100% because Pythium can go from, it can go to monocots, it can go to dicots. It doesn't care. There's 300 species of that pathogen, and it is pretty ubiquitous all over the world. All right, so switching kingdoms to bacteria now. So there are ones that we have in this state that are seed-borne, and there's ones that we have that are vectored by insects. So angular leaf spot, bacterial leaf spot, seed-borne, cucurbit yellow vine disease, and bacterial wilt are vectored. So on the left-hand side, this is bacterial leaf spot. Just looks kind of these insignificant little spots, kind of yellow halos. Sort of looks like Cercospora leaf spot a little bit. And then angular leaf spot is the description is in the name. These dark brown lesions that are more angular. One way to kind of differentiate fungal foliar diseases from bacterial diseases, fungi are going to have sporulation on the other side of the leaves. That's something bacteria cannot do. So there's be a bunch of little spores kind of hanging off the underside of the lesions. And another thing is that bacteria have to grow kind of restricted by the veins of a leaf. So a fungus can kind of grow over these main veins, whereas bacteria have to kind of stay within them. So if you see a lesion that's just like doesn't really care where it's growing at, it's probably a fungi. But if you see these where they're kind of in between the veins only, that's probably a bacterium. So that's just kind of a good diagnostician's trick right there.
Is the bacterial leaf spot, is that an early stage of it or a later stage?
This right here,
Yeah.
it looks like a mild case at a later stage. I know, that was probably one of the clearest photos I could find of it, but this doesn't look that aggressive, to be honest. This looks aggressive. This looks bad.
A leaf spot would get a lot worse than that.
Yeah, it can. So a little easier disease cycle this time. And again, this is a tomato. I understand it's not a cucurbit, but I'm limited by my free images. So we have our infested seed. You plant it, and at this point, you're not going to see any symptoms. But as these plants mature, you're going to start seeing that spotting in the leaves of those angular leaf spots. And then bacteria, way better than fungi, have a lot of secondary disease cycles. Bacteria replicate really, really fast. And so this bacteria can spread throughout your field. And it doesn't have the ability that fungi have to kind of like catch wind and blow away, but rain splashing, physical movement, any way, wet leaves are moved around and touch each other is going to spread this bacterium. Then at the end of the day, I don't know how many of you save your seed, but if you do, that seed's going to be infested. So when you use that seed in the next season, you're just going to have the same symptoms, right? It can live within the seed coat. So obviously, don't plant infested seed. Try to buy some disease-free seed. That one is very obvious. There's resistant varieties to both of these bacterial diseases, so you can actually find in your seed catalog that there's angular leaf spot resistance, there's bacterial leaf spot resistance. And then a good tip, depending on the size of your operation, could be to rogue out these disease plants because, every seed in your packet's not necessarily infested, so this may be a one-off or a 10-off, of your plants, and maybe you can rogue out these plants and save the rest of yours. That's just going to kind of be based on what you see in your field. Just like for fungal pathogens, like foliar fungal pathogens, avoid overhead irrigation because that rainwater is going to splash the bacterium that is just oozing out of these leaf spots all over the rest of your field. And don't work in wet fields that are contaminated with bacterial foliar diseases because you are physically moving it around in your field if you do that. And then this may or may not persist in the soil. I could not find a clear-cut answer on this, but for these diseases, tillage may be useful and crop rotation may be useful. I think the primary way these diseases are spread is by seed, but this may also be helpful in a small sense. I'm not sure. Okay, so now our vectored bacterial diseases. So bacterial wilt is going to be vectored by cucumber beetles, but then there's also one called yellow vine disease, and this is vectored by squash bugs. So two different bacteria, two different diseases, two different vectors. There's really not much to say about them. They look the exact same. A plant just kind of collapses. So this is kind of looking at, now I do have cucurbits. This is looking at the cucumber beetles. So let's start with the beetles. They're in your field. They're all carrying the bacteria within their guts. And so they start feeding on plants, and they start transferring this bacteria into the vascular tissue of your plant. And over time, due to feeding and just bacterial replication, it's going to plug up the vascular tissue, and your whole plant's going to collapse because it's not able to translocate water or anything. And then your whole plant's going to collapse like that. It might happen pretty rapidly. And then your cucumber beetles are going to be all happy and satisfied and kind of just go sleep under all of your crop debris or your plastic. and they will overwinter there until the next year and pop out and then completely, restart this disease cycle. And so obviously we have to manage the insects. If you're going to have any kind of control, you have to have insect control. So using insecticides, if you can, using netting, if you're doing organic production and you have less effective products, using netting to actually keep these insects out is going to be your best bet. You can use trap crops to actually get these insects to go to these other non-cash crops that you have. So using like Hubbard squash for squash bug is a good way to kind of keep them off some of your more cash crops that you have. Crop rotation will work for squash bugs. I just talked to Amanda Bachman. She said rotation will work for squash bugs, but not so much for cucumber beetles, because cucumber beetles can fly up to like 500 miles, whereas squash bugs are more like walkers. So crop rotation may or may not work depending on which bacterial wilt you're dealing with. And then host resistance. Again, if you have it, use it. It's your silver bullet 99% of the time.
Did
Yes.
you say that in the cucumber beetles, they hide underneath the plastic also?
Yeah, I think I'm not an entomologist, but I think what they do is like anywhere they can hide under, whether it's plant debris or your plastic, they will find some nice warm place and hide out for the winter.
So what's the best way to go about getting rid of them?
Ooh, from the soil?
Spray when you see them, or a prevention plan?
I don't know the best answer to that. I don't. I would honestly say that you want to use a contact insecticide sort of like as you see them in their younger stages. That's just kind of my best guess, but because as soon as they get in their adult stages, they're harder to kill off. So earlier in the season is the better.
The best way I found out, as soon as I can roll my cucumbers, I go cultivate and spray.
Yeah. Whatever works, whatever method you can to get rid of them, keep the numbers down. And then, so if you can rogue out your diseased plants, if possible, probably your cucumber beetles are already full of this bacteria, but potentially one that doesn't have this bacterium in its guts can feed on this infested plant. You're not going to get anything out of this plant anyway, so it makes sense to kind of remove this bacterially infested plant off of your farm. Lastly, and real briefly, viral diseases, which are also vectored by insects: squash mosaic virus and cucumber mosaic virus. So squash mosaic virus is cucumber beetle vectored, and I think there's some other beetles out there that can vector them. I don't know them off the top of my head. It's also seed-borne, so it can come in your seed, and it may overwinter in other perennial weeds. So this is just kind of a trifecta of trying to keep this off of your farm. So it comes in your seed, it comes on bugs, and it's in your weeds. Same is kind of true for cucumber mosaic virus, except this one is aphid vectored. And it also may overwinter in other crops that you have. So the host range of cucurbit mosaic virus is a lot wider than it is for squash mosaic virus, which mostly stays within cucurbits. So just like for bacteria that is vectored by insects, roguing infested plants, buying disease-free seed, controlling vectors, host resistance, all are going to be great strategies to control these. Again, viral control is a lot harder than bacterial or fungal control, so you really kind of need to have a good plan kind of for the next year, because there's not really much management you can do other than just trying to get stuff that's disease-free starting out. And so what I didn't give you is a lot of practical information like spray rates, chemicals, stuff like that. And so what I want to direct you to is the 2023 Midwest Vegetable Production Guide. So in there is a bunch of researchers coming together and like combining efficacy studies for different products. In this case, this is for diseases, but there's also insect pests. And this is available for free online. You can just download it. And they have all these efficacy tables here. So this is for fungi. And they'll kind of say, oh, this is very good control, fair control. And it'll give you a kind of starting point. So if you see that disease, you can sort of plan ahead for the next year. And this is what I'm going to use in my own field trials. So I'm not going to tell you to use something that I'm not going to use myself. But yeah, so they also have one for insects as well. And with that, I am new, so my lab's website is coming. But if you want to email me, please do. If you have any disease questions, I still need to see a growing season in this state. I'm on Twitter, too, if you guys are. And, yeah, if you have any questions, feel free to ask.
I have the powdery mildew gets in my high top. And I know now it's really important to try to get some of the varieties that are more disease resistant.
Yes.
Because it'll start in my peas, my snow peas and my sugar snaps, and the minute I see that coming, I know I gotta get them out of there.
I will say, though, that the powdery mildew that's causing powdery mildew in your peas is not the same powdery mildew on your cucurbits.
Really?
Yes.
That's interesting, but I had 3 varieties in there, and one of them got hit really bad, and the next one, nothing. So, I won't plant that one.
It could just be like the time of the year for powdery mildews. Yeah, powdery mildews are obligate pathogens. So like what that means is you can't, like for a lot of microbes, you can grow them in culture. For powdery mildews, they're obligate pathogens, and they often evolve with their host specifically. And so they can't like genetically recognize other plants. They can only recognize their host. And so there's thousands of hundreds of powdery mildew species out there. Like the powdery mildew on grape is different. The powdery mildew on, beans or peas is probably different. So yeah. It's a fun fact. Anybody else? All right, well, thanks for coming out.
But thank you.
Thank you.